And I won't say good morning or good afternoon because I'm sure we're scattered about the globe. I'm so happy to see everyone's names. There are a lot of names here I recognize. For those of you who don't know me, I'm Steve Finkel. I'm a professor here at the University of Southern California and also a member of the C-DEBI team. And I'm really happy to have you all join us for the first of these seminars during the new year. It's my pleasure today to introduce Jesse Colangelo, who's been taking a question that's really hard and has made it even more difficult. And it's been really fun watching him tackle what I just think is a brilliant experimental system, a really novel experimental system, and something that I think you're all going to find really fun. The way it's going to work today is Jesse's prepared about a half-hour presentation. He has very graciously agreed to be interrupted if necessary or as necessary. So what's going to happen is, as Jesse's doing the talking, I will monitor the chat. And if there's something that comes up that I think would be really good to interrupt him about, I will. If you think it's super important that he be interrupted, indicate that to me. And then we've left a good block of time at the end for some robust questions and answers. And then we will honor everyone's time and have a hard stop at 1:30 Pacific time. So Jesse is a NASA astrobiology fellow and spends time both at Colorado School of Mines and CU Boulder. And I'll let him introduce his amazing system. And Jesse, I'm looking forward to your presentation.
Great, thanks very much, Steve. And thanks everybody for tuning in here. I really do appreciate your attention. So today I'll be talking about some experimental evolution experiments that I've been working on for the past few years, including during my time as a C-DEBI postdoc. This work I'm going to tell you about is primarily concerned with what stable isotope biosignatures can tell us, both about the environment and the organisms that utilize these elements. So most elements have multiple isotopes, and what makes an isotope is a different number of neutrons. So each isotope of a given element has the same chemical properties, but a different mass. Biological processes tend to favor utilization of light isotopes of a given element. And probably the most well-known example is that of plants and other photosynthetic organisms preferentially using carbon-12 carbon dioxide, such that their biomass is depleted in carbon-13 and enriched in carbon-12 relative to the reservoir of carbon dioxide that they're drawing from. Microorganisms that use oxidized sulfur do the same thing. Unlike carbon assimilation during photosynthesis, the reduced sulfur that they produce is not assimilated, rather it is excreted. But in both cases, the ratios of heavy and light isotopes in the reaction substrate and the product can be quantified, and the preferential use of one isotope over the other is called fractionation. In the case of sulfur, both the reservoir sulfate and the product sulfide are subject to mineral preservation, and as such can serve as biosignatures of sulfur metabolism over very long periods of time. A number of environmental factors are known to influence the extent to which microbes will produce isotopically lighter metabolic products like sulfide. And these include things like temperature and pH and concentrations of substrates and products of reactions that are biologically catalyzed. And as a result, various interpretations about the environment through time have been made for measurements of isotopes recovered from old rocks. Here you can see a data set of the isotope composition of sulfides and sulfates recovered from rocks of different ages. The mineralized sulfides are the black bars, and the blue lines is the mineralized sulfate. The mean difference in composition between the two, you can see here in the top right, and that there's an expansion in fractionation as we move from deep time to the present. This data, coupled with growth experiments showing that isotope fractionation is diminished when sulfate is in low concentration, one interpretation is that the global ocean was sulfate depleted compared to modern concentrations for much of early Earth's history. There's a number of other geobiological interpretations that have been made on the basis of sulfur isotopes, including determining the export of marine organic carbon during the Phanerozoic, constraining the extent of continental shelves, setting a lower time limit on the origin of the sulfate reduction metabolism, and determining the timing and progressive oxygenation of the atmosphere. These interpretations of geochemical events and processes through time on the basis of sulfur isotopes make a few assumptions. And among these is that this black box of microbial activity and its associated isotope fractionation responds to the environment, but the black box itself is constant. And more specifically, this suggests that sulfate-reducing bacteria have been fractionating isotopes in response to the environment in the same way over geologic time. But is it instead possible that some of these differences in fractionation that are attributed to the environment are instead attributable to the organisms? For example, is it possible that the increase in fractionation observed between sulfate and sulfide-bearing minerals is not due to changes in global oceanic sulfate concentration, but instead due to adaptation of the organisms that are responsible for that fractionation through time? So my work focuses on sulfate-reducing bacteria, which primarily occupy marine sediment environments. And so the projects that I'm going to describe each envision one of the challenges, or at least a change that microbes experience as they are buried in progressively deeper sediments. The first of these is the loss of sulfate as an electron acceptor. This is a depth profile that's typical of coastal and continental shelf sediments, where the majority of sulfate-reducing bacteria are active. And you can see that the sulfate is quickly depleted with depth. The second challenge is the loss of electron donors, which for heterotrophic sulfate-reducing bacteria means labile organic carbon. And this depth profile, again, is typical of sediments inhabited by sulfate reducers. And you can see that the easy-to-eat organic carbon is quickly depleted, leaving only the difficult or impossible to eat organic carbon. And then the third challenge considers transitioning from a planktonic growth environment to particle-associated growth. And this is not a challenge encountered by contemporary sulfate-reducing bacteria, but likely was a challenge when the oceans transitioned from anoxic to oxygenated sometime in the Paleozoic. So in each of these challenge experiments, I want to address whether the microbes can survive the selection pressure, and if they can, if there are isotopic consequences to any adaptation that has occurred. Okay, so the first project I want to tell you about is a piece of a larger project looking at an array of adaptive properties of centrify. The experiment was to force a sulfate-reducing bacteria to grow in obligate centrify with a methanogen. No sulfate was provided, and so the sulfate-reducing bacteria has to ferment its organic carbon source, and the methanogen makes use of the products of fermentation for its own growth. Notably, these sulfate-reducing bacteria methanogen centrifuges are known to be present in marine sediments below the sulfate-methane transition zone. So replicates of these co-cultures were transferred for 1,000 generations, serially transferred. And from these 1,000 generation co-cultures, I tried to isolate clones that were still capable of utilizing sulfate, and then interrogated their growth physiology, resequenced their genomes, and looked at the fractionation of sulfur isotopes, again, when they're grown in the presence of sulfate. And we can compare these characters to those of the ancestral strain because we can resurrect the ancestral strain from stocks. Okay, in answer to the first of my questions, we were able to isolate clones capable of sulfate reduction when they were grown in media containing sulfate, which confirms that some were able to survive the selection pressure of electron acceptor depletion, at least with the help of a centrify methanogen. Notably, this was true from all the 1000 generation co-cultures, but it was not true for the majority of the clones that were still capable of growing on sulfite, indicating that some of the upstream machinery involved in sulfate reduction was likely broken. We resequenced the genomes of a subset of these organisms that retain sulfate reduction capacity. And you can see here a snapshot of the gene variants observed when compared to the ancestral genome. Each resequenced clone is listed 3 times at the base of each of the 3 columns, and each column is for a different sort of gene variant. Some of these gene variants make the gene lose function entirely, and others are more likely to change its function but not end it. Unsurprisingly, many of these genes have unknown function, but those that do have predicted function are primarily annotated as playing a role in transcriptional regulation, in ion transport, or in sulfur catabolism. Several of these gene variants were observed between multiple sulfate-reducing clonal lineages, and these sulfur catabolism genes include a sulfate transporter, a general sulfatase enzyme, the sulfide reductase gene that catalyzes the last step in sulfate reduction, and a lactate transporter. Okay, so having identified clones capable of sulfate reduction and their gene variants, the next thing to do is to look at how well they were able to grow compared to their ancestor under conditions requiring sulfate reduction for energy conservation. So you can see that these evolved lineages are diminished both in their growth rate and their yield compared to the ancestral strain, presumably as a result of their diminished capacity for sulfate reduction. Towards addressing the question of whether their isotope phenotype was also altered, I measured the isotopic composition of the sulfate and the sulfide from batch cultures. And this right-hand figure here shows the sulfur isotope fractionation plotted against cell-specific sulfate reduction rate. And you can see that both fractionation and cell-specific sulfate reduction rate are diminished compared to the ancestor in all but one evolved lineage. So this answers in the affirmative the second question I had hoped to address as to whether there are isotope fractionation consequences to adaptive evolution. But I wanted to explore this relationship a little more deeply. So this particular relationship between fractionation and cell-specific reduction rate is one that has been examined before, both in batch and continuous culture, and demonstrates this exponential inverse relationship. That is, at these very high reduction rates, fractionation is low, and at very low rates, fractionation is high. And so the expectation is that when reduction rate decreases, fractionation should increase, which is the inverse from what I saw in my batch culture data set. So to look a little bit deeper, I took the evolved lineage with the greatest apparent diminishing of fractionation and grew it, and separately the ancestral strain in continuous culture at numerous growth rates. And what I found initially surprised me, which was that the fractionation exhibited by the evolved lineage was not consistently diminished compared to the ancestor, but its being either greater or lesser was dependent on the cell-specific reduction rate. So I used a thermodynamics-based metabolic model to fit this data set. And based on the parameters of that model, I think this trend reflects which factors are actually serving as the bottleneck to sulfate reduction for this evolved strain at different reduction rates. So in summary, for this experiment, which subjected a sulfate-reducing organism to electron acceptor depletion, the organism was able to survive the selection pressure, and there were isotopic fractionation consequences to that adaptation. This next experiment was designed to address the same question of isotopic consequences of adaptation, but here the selection pressure is depletion of the labile organic carbon that serves as an electron donor. The experimental design here was initially modeled after a recent antibiotic resistance experimental evolution platform. And here, rather than using Balch tubes or chemostats, The idea is to grow the organism of interest on this plate that spatially provides either both edible and inedible organic carbon, or after that area has been colonized, only inedible carbon sources. One advantage to this approach is that it allows for resolution and separation of successful populations that can then be interrogated individually for the genetic means by which they have adapted to utilizing a novel organic carbon substrate. In this case, the organism was a sulfate-reducing bacteria, and so these plates had to be operated in an anaerobic chamber. Before I could start the experiment I had in mind, I had to identify growth conditions conducive to the sulfate reducer growing in low density agar on top of a high density agar containing the organic carbon. And this turned out to be much more difficult than I anticipated. And for over a year, I worked towards identifying these conditions, and a subset of the many plating conditions that did not facilitate complete colonization are shown here on the left, and each of these plates was incubated for a month. Even after eventually meeting with some success getting complete colonization of large plates, it was often the case that I could not replicate complete colonization between replicate large plates. And so shown here on the right are two plates, both 10 centimeters by 25 centimeters, and each one divided into two sections. The first plate is an example of the organism colonizing the complete plate surface as expected, since both sections of the plate contain edible organic carbon. The second plate is one of many experimental plates where one section contains both edible and inedible organic carbon, and the other section contains only inedible organic carbon. And in this case, it does look like there has been some successful colonization of the area of the plate that represents electron donor depletion, but at the same time, the area of the plate that definitely should be colonized is not. Both of these plates were incubated for 2 months. So due to the slow growth and physical restrictions on how many plate conditions I could work with at once in the glove box, I decided this would be a better experimental approach to resume after I have established my own lab or secured a longer term position. And so I decided to try and address the same problem with a higher throughput, more traditional experimental evolution design. So that more traditional experimental design utilizes small volume aqueous cultures in replete media that can be successively transferred. So here, all the cultures are grown in media containing both edible and inedible organic carbon molecules and are serially propagated for 35 generations to give them time to possibly figure out how to eat some of these inedible carbon sources. After this period, the cultures are interrogated for growth and compared to parallel cultures grown only on the edible organic carbon sources. So in the cases where the area under the curve is greater for the cultures grown with both edible and inedible organic carbon, I assume that augmented growth resulted from the utilization of previously inedible sources, and then another round of cultures were grown on new media containing only the previously inedible organic carbon to confirm that some novel capability had emerged. It shows these organic carbon sources on the left because each of them is used as an electron donor by some heterotrophic sulfate-reducing organism. And because of the higher throughput capacity of this experiment, I employed two sulfate-reducing organisms of different character, one heterotrophic incomplete oxidizer, the model organism Disulfovibrio vulgaris, and one auto or heterotrophic complete oxidizer, the Sulfobacterium autotrophicum. I expected if either organism was likely to survive this depletion of edible organic carbon, it would be Disulfobacterium autotrophicum, which has a much larger genome and is already capable of more versatile metabolism. Okay, this data set is from the first set of cultures which were grown to establish a baseline of the organic carbon compounds that each of the sulfate reducers are capable of using. So basically, I'm just confirming what you can find in the literature, but sometimes it's good to check. So each of these plots looks at cell growth over time in media with different sources of organic carbon. The red lines are cultures with no organic carbon, and the black points are replicate cultures containing the named carbon source. Note that the y-axis per plot is scaled to the data range. You can see here that this organism can use pyruvate and lactate for growth, but not the other carbon sources intended for this experiment. These plots show the growth for the other sulfate reducer, Disulfobacterium autotrophicum, subject to growth on the same set of carbon sources. And you can see here that this organism is also capable of using both lactate and pyruvate, and additionally fumarate, ethanol, proline, and possibly a few other organic carbon sources very poorly, like glutamate. Okay, so these next two sets of plots show growth data for the two sulfate reducers grown in media containing both organic carbon sources that they were able to use at the start of the experiment, and also carbon sources that they were unable to use following 35 generations of growing in these mixed medias. And again, I'm looking for cultures that have a greater area under the growth curve when they're grown with both the edible and the inedible carbon compared to the area under the growth curve when grown with only edible carbon. As an additional control to show that all of these cultures are in fact limited by organic carbon availability, I also grew the organism with double the concentration of lactate and pyruvate, and you can see that those cultures did result in a greater abundance of cells. The other thing that you can see here is that Disulfovibrio vulgaris did not demonstrate augmented growth in any of these mixed carbon source cultures, and so presumably would not be able to adapt to the selection pressure of organic carbon depletion. These plots show the growth for the other sulfate reducer, the Disulfobacterium autotrophicum, which I previously determined was capable of growth on lactate and pyruvate and ethanol and fumarate. And so those are provided as edible carbon sources in tandem with combinations of the other inedible carbon sources. These growth curves are from cultures that have been grown under these edible plus inedible conditions for 35 generations. And in contrast to the results from Disulfovibrio cultures shown in the last slide, it does look like a few of these replicate cultures are exhibiting augmented growth in some of these media, specifically in media containing aliphatics and methylated carbon molecules. However, you'll remember from a few slides ago that the final step of this experiment was to try and grow the organism solely on any individual, initially inedible carbon source. And I just decided to save the slide showing the data and just tell you that none of these cultures were able to grow with any of these aliphatic or methylated molecules as their sole source of organic carbon, which answers in the negative whether these organisms would be able to overcome the selection challenge of electron donor depletion. So in summary for this experiment, where I attempted to look at survivability and isotopic consequences of the selection pressure of organic carbon depletion, I was not able to isolate any individuals that were able to adapt and overcome this selection pressure. The final experiment I'd like to tell you about imposes a selection pressure for adhering to particles. This selection is meant to be a very simplistic model to recapitulate what may have happened to sulfate-reducing bacteria several 100 million years ago when the global ocean was oxygenated and oxygen-sensitive organisms were relegated to anoxic niches, including sediment environments, where the majority of microbes are thought to live in some physical association with sediments. To my knowledge, all experimental work with microbes that is used to establish relationships between environmental variables and isotope fractionation uses aqueous cultures. And at least for organisms that reside in sediments, a physical environment more representative of their habitat may be more appropriate for these experiments, which is what I'm trying to address here. So the design for this experiment was to grow a sulfate reducer in serum vials with 50 micron sterile resin beads, which are meant to emulate sediment particles. Each successive transfer from those cultures to inoculate new cultures used washed beads as inoculum rather than the aqueous phase. And we expected this serial transfer to enrich for progressively larger proportions of the population adhering to the beads. After 35 generations of growth, we interrogated these populations for whether they were in fact particle associated and how this association influenced growth and isotope fractionation. So in answer to the first question, which is a bit self-evident, both from observing growth in successive experimental cultures and also from the fact that we see sulfate reducers in contemporary marine sediments, the organism was able to adapt to the selection pressure imposed. And you can see that in these images, which compare the appearance of the resin beads at the start of the experiment from the first transfer and the resin beads after generation 35. You can also see that while these beads are certainly colonized, the cells are also not making a thick biofilm, which is important for our objectives to quantify both growth and isotope fractionation parameters, both of which require that we can accurately enumerate cells. So the ideal outcome of our serial transfer of resin beads would have been a transition of the population from entirely planktonic to entirely particle associated, but that's not quite what we found. Instead, what we found was that while the majority of the cells in the culture were particle associated, as much as 40% of the cells at the end of the 35 generation culture were still planktonic. So in order to make calculations for growth parameters for the particle associated cells, I had to make this assumption about how those cells and the planktonic cells were each using sulfate for growth. And specifically, the assumption that I made was that biomass generated from sulfate consumption, or yield, was the same between the planktonic cells in the positive control cultures and the planktonic cells in the experimental cultures containing resin beads. Okay, so here I show the growth parameters, how they varied for the experimental culture cells, both planktonic and particle associated, compared with the control cultures. The yields from the bulk experimental cultures were actually very similar to the planktonic controls, though a tiny bit smaller for the particle associated cells. However, the growth rates of both the planktonic and particle associated cells were smaller than the planktonic control cells. These two parameters, growth rate and yield, are combined to generate cell-specific sulfate reduction rates. So both these rates and the fractionation of sulfur isotopes between the substrate sulfate and the product sulfide are plotted here on the right, both for the control culture and separately for the particle associated and planktonic cells from the experimental cultures. There were 5 replicates of each control and each experimental culture. While individual cell-specific sulfate reduction rates can be teased out of the data for the particle-associated and the planktonic cells in the experimental cultures, there is not a good way to tease apart the fractionation that can be attributed to each population. And so each is plotted as though they fractionate equally using the bulk culture fractionation value. I've also included the modeled reduction rate fractionation relationship as this dark blue line. And at these high cell-specific reduction rates, previous work suggests that fractionation is small and almost constant. However, you can see that the experimental values for the particle-associated cells do not fall onto this modeled line. And my interpretation of that deviation is that some changes associated with adaptation to particle-associated growth are influencing fractionation in a manner that cannot be attributed simply to changes in growth physiology that are driven by environmental factors and would plot somewhere along this line. Okay, so I've covered a lot in the past few slides, and I want to briefly recap. I undertook these projects to explore whether the organisms responsible for fractionation of isotopes might play a role beyond simply responding to the environment. And the projects I undertook looked at sulfate reducers' response to electron acceptor depletion, electron donor depletion, and transitioning from planktonic to particle-associated growth. And you can see here a quick reminder of which selection challenges the microbes were able to overcome and which of those resulted in a change in isotope fractionation. What I have not done as part of this work is developed a new model by which to interpret isotope biosignatures that would incorporate both biological and environmental contributions to fractionation. I do think that this work provides evidence that working towards such a model is a worthwhile goal for better interpretation of isotope biosignatures. And if we could develop such a model, it might be able to provide a complement to molecular phylogenies in teasing out the timing of specific adaptations or evolutionary events. What I would like you to remember about this presentation is that the biology that is responsible for isotope fractionation changes along with changes in the environment, and that those adaptations can influence fractionation in addition to the changes wrought by environmental change. Okay, so my last slide is just some acknowledgements. I didn't do all of this work on my own. I had lots of help in many aspects of these projects from PIs and research scientists and graduate students and undergraduates who are listed here. And I would also like to extend my thanks to the C-DEBI Executive Committee and staff and entire community. I have enjoyed and benefited immensely, personally and professionally, from the expertise and scientific camaraderie of this community. And I will just leave it on this slide, and I'd be happy to take any questions.
All right, thanks so much, Jesse, for a really fantastic presentation, and thank you for staying to time. I appreciate it. Yes, there's applause coming in the chat. So let's wait a moment. Okay, so we have a question from, I think it's Myrna Jacobson. Do you have a feel for the pore space between the 50 micron sterile resin beads? This might influence the attached, ooh, it's streaming in too fast, the attached versus free floaters. And she enjoys your great hunk of work.
Thanks very much. Do I have a sense of the pore space? No, but I know that we could quickly calculate that. The distribution of sizes is pretty small, and so they pack like regular spheres. And I can't remember off the top of my head what that ratio turns out to be, but it is substantial. And that is why we washed the resin beads prior to using them for inoculum in hopes of removing as many of those free floaters as we could prior to transferring into the next serial culture.
Great. Next question is from Arcady. With regard to the first half of your talk, propagating 1,000 generations under electron acceptor depleted conditions, thinking about the genetic basis for adaptation, how divergent were the evolved strains relative to the ancestor? Yeah, this is also, as you can imagine, a question I might have asked. With sufficient divergence, are there signs of any relaxation in selection or any positive adaptive events?
Yeah, so as I preceded my contribution to that much larger project, that work has actually been done. And so I was primarily focusing on clones that we could pull out that had maintained the ability to reduce sulfate because I'm interested in sulfur isotope biosignatures. But there are lots of individual populations that have been recovered from those 1,000 generation co-cultures that have been genomically interrogated. And how do I? There's a paper on it. Who is the first author? I think Serdar might be the first author on it. I can get back to you if you want to email me if you actually want to read the paper. But there is quite a bit of divergence, and the paper goes into depth for a slew of signal transduction genes, which were the things that really jumped out at those authors as being quite divergent. The sulfur reduction genes were more often than not wiped out upstream of sulfide, so non-functional, which is why you didn't see any of them in my gene variant plot, because those cultures were no longer able to grow on sulfate.
Great, thank you. The next question is from Elliot. With respect to the electron acceptor depletion experiments, have you considered whether the evolved strains have mutated DSR or APR enzymes that may affect isotope effects independent of cell-specific sulfate reduction rate?
Yeah, absolutely. So that is what this body of work is getting at, is that there is almost certainly adaptation within the sulfate reduction pathway and within everything that interfaces with that pathway that will affect fractionation. I didn't do any sequencing from any of the electron donor depletion experiments because none of those things survived. So I wasn't able to do any isotopic characterization that then would be more interesting to look at the sequencing. But certainly, I expect that those genes are susceptible to mutation under the right selection regimes that would influence isotopic fractionation.
Yeah, that's cool. Myrna's following up. I mean, yeah, this question of the attached versus the not attached and the actual space is really important. So she's following up with, it's essentially, do the microbes know what kind of a space they're attached or not attached? And for example, she says in a mud environment versus sand environments, there's a perception of attachment is going to be different. And it's based on, the water. I mean, I'm just going to editorialize a little bit as well, Myrna. There's the packing density, there's the porosity, there's what's the ratio of solid to liquid, how dense is the solid material. So if you had tweaked the knob a little bit and used different particles, do you think you might have gotten a different result? I guess is maybe where Myrna's going and what I'm sort of thinking about as well. It's hard, right? No, I don't think anyone's ever done this before.
I just don't know. I mean, it wouldn't be ridiculous to try and do exactly that. So, if you have more pore space, then you have a larger fraction of cells that remain, in the sediments that are not, in fact, sediment associated. And so, is it easier or harder to wash them out? I don't know. I guess I don't have any sense of the physics of how good the washing is actually doing the job that I think the washing is doing, and whether that job would be more efficient if the beads are smaller. I guess if the beads were larger, it's more efficient to wash out the things between them.
I think that, if you were going to go this way, something that could be done immediately would be, and it's easy for me to suggest it, of course, is, reviewer number 3. If you use different size beads, and if you actually just counted the number of cells planktonically, if they cared whether or not you had more or less space between the particles, if your planktonic cell counts never changed, that may or may not tell you anything about the attached. But if they do change, I think it definitely tells you something about that. So that would be a quick and dirty way to see if they actually care, with one change of the condition.
The concentration of planktonic cells did decrease after being transferred via beads.
Yeah, so then it becomes this wonderful competition between would they rather be surface attached and there's something bad about coming off the surface versus why, are they just not coming off, and how are they sloughing into the planktonic milieu?
It looks like it's to their advantage to find a way to slough off because their growth rate was slightly higher than when they were attached to the beads, but that's only true until the next selection regime of me transferring the beads wipes them out.
Yeah.
But yeah, I think they would rather not be on the beads.
Oh, that might well be. Let's see. We've got the next questions from Will. He thanks you, Jesse. If you were able to normalize to the number of enzymes rather than the number of cells for comparing the J to epsilon values in the planktonic versus particle associated, do you think the rate / fractionation relationship would again be negative, or would it still be positive?
That's a great question. I don't know. We didn't do any quantification of enzyme expression. And certainly that could be another explanation for what that deviation is that we are just not looking at the right metric. The metric should be the enzymes because enzymes are in fact differentially expressed.
And if you were to even quantify, would you do that by gene expression and infer more or less, or would you actually be able to count the enzymes directly through some biochemical method?
Oh, I would almost certainly go the molecular route rather than the
Will says
biochemical method.
proteomics, exclamation point. Yes.
Yeah.
Okay, great.
Yeah, it would be interesting just to do a back of the envelope calculation to see what those enzyme concentrations would need to vary by to account for those differences, which is not something I'm going to do on the fly, but maybe I'll do right after this talk.
Great. Yeah, and Myrna is suggesting you can do experiment, things with labeled substrates and such. Although looking at flux might not tell you, because you could have same or different numbers of enzymes and have similar or different fluxes. So it could get weird very quickly. Any other questions? While we're waiting for to see if there's any more questions, I did want to ask you, Jesse. I'm glad you ended on this slide. When you look at the middle experiment, I agree, Will. The weirder is the better. When you look at the slide, we've got the one experiment that didn't particularly work, which is actually, of all the experiments I know we've been talking about over the last couple of years, this is the one I was thinking was going to work. So when it doesn't work, there's a bunch of reasons. We don't know what they are right now, but one possibility is simply that there are or not enough cells.
Right.
To provide enough genetic diversity. So when you think about your three platforms that you've created here, the electron acceptor depletion, the electron donor depletion, and now this planktonic particle experiment, do you have a feeling for the relative numbers of cells, or can you share with us, what are the relative number of cells per experimental unit, just so we can get a feeling of, is there a big difference in the number of genomes that are simply available to undergo mutation and natural selection?
I can't remember the top of my head how many cells are in like a square centimeter, but I can tell you that in these aqueous cultures, which are primarily done in serum vials, concentrations of cells get into the billions per vial. So between billions and tens of billions of cells per vial, and then you go through several transfers of those. And are there a comparable number on these plates? I'm embarrassed. I can't remember how many cells there are in a square centimeter of, colony. But again, we could back of the envelope that. It's got to be not too far off.
I agree. And your typical colony has around 10 to the 8 cells, if you can see it. So you imagine a square centimeter worth of that. It's a lot of cells.
In that case, then there's many more cells when they are successful in colonizing the plates.
Yeah, and then what's fascinating is the number of cells in the bead experiment might actually be a little lower.
Right.
Yet it worked.
Yep.
Yeah. Which, and again, it's, you only need, really depends how many different alleles there are that are going to help you out, and you can do again, there's a lot of arithmetic you can do to decide what your minimum is. Obviously, it worked. Well, actually, I guess maybe my last question was, my note was, how do you tell the difference between physiological adaptation and genetic adaptation? So could it just take 35? Can it take 35 passages just to physiologically adapt to the new environment, and they're actually genotypically identical?
Yeah, that's absolutely possible. And so we still have these things, and I'm planning to sequence them, but I haven't yet. And until then, we can't distinguish between the two.
All right. Do we have any more questions out in the audience? And again, I apologize. We're in a webinar environment where I can't see anybody if they're raising their hands or nodding or shaking their heads. You're getting some nice comments, so I'll let you read those.
I can't see the chat window.
Oh, you can't see the chat window. In that case, you're getting. Then in that case, we'll say maybe Matt can figure out a way to cut and paste it, and we'll actually, yeah. Can we do that, Matt? Can you like grab the chat and just email the text to Jesse so we can see all the questions and who asked them?
Yeah, I think we can do that.
All right.
Yeah.
Thanks, Rosalind. Yeah, that would be really nice because there's some nice suggestions in here.
Yeah, I think Rosalind will have to do that.
Okay, that's fine. We'll do that for you, Jesse, and not just because people are saying nice things about you, although it is nice. I'm a big fan of when someone's saying nice. It's nice that people are saying it in front of you. Anything else right now, folks? Anything anyone else would like to muse on? We've got a few minutes. No one minds ending early, I'm sure. But we are here, folks want to do so. All right, I guess then I'm Jesse. On behalf of all the attendees and of the C-DEBI leadership, I just want to thank you for what was a really terrific presentation summarizing some very thought-provoking work. You know, we've been talking for years, and you were definitely pushing the envelope, trying to merge two really hard fields. And it's a, you know, I really like seeing those check marks. My only comment would be maybe make them green. Or some other bright color, because the amount of work that went into those black check marks is really extraordinary. And you know, and I'd make the X's smaller and the checks bigger, because it really is it really is beautiful that, you know, 2 out of 3 ain't bad.
Great.
Okay, and Will's just following up for the chat part. Oh, cool. Thank you very much, Will, for that. That's really great. So we'll get that passed on. All right, in that case, everyone, thank you for attending. Thank you for your questions and your attention today. Thank you to Rosalind in particular for all of your help with this, and Matt for joining us today. You know, none of this happens without amazing guidance and thoughtfulness from Rosalind, and we just could not do anything without you. So I'm very grateful. And yes, we're all going to get vaccinated. Well, I live in Southern California, so I'm way down the list. But be well, take care of yourselves, and we'll see you next time, everybody.
Thank you. Bye, everyone.
Bye, everybody. Great job, Jesse.
Thanks, Steve.